Hello and welcome to today's video lecture on urine formation. Dear students, urine formation occurs in three steps. Step 1 is glomerular filtration, followed by tubular reabsorption, and the last one is tubular secretion. As we know, nephron is the basic structural and functional part of kidney. All these three steps of urine formation, that is the glomerular filtration, reabsorption, secretion, they happen at the level of nephron. Nephron consists of a Malpighian body and the tubule. The tubule can be further divided into three different parts, that is PCT, loop of Henle, and DCT. PCT is proximal convoluted tubule, loop of Henle, and then finally there is DCT. DCT is distal convoluted tubule. Process of filtration happens in glomerulus. The filtrate after filtration enters the tubular part of the nephron and passes through the PCT, loop of Henle, and DCT, whereby reabsorption and secretion happens. As far as nephron is concerned, there are two different types of nephrons. That's cortical nephron and medullary nephron. Cortical nephron has major part in cortex of kidney, and they are 3 upon 4 of the number in a kidney. Medullary nephrons, they are long nephrons with long loop of Henle deep into the medulla of the kidney. They compose 1/4 in terms of number in a kidney. The network of blood capillaries around the tubule of the nephron helps in reabsorption and secretion. Here you can see in the figure, red colored vessels show arterial part and blue colored vessels show the veins part, and then there are small capillaries in between. Here, the long loop of Henle with capillary network around it is called vasa recta, and vasa recta is present in case of medullary nephron. In today's video, we will be covering the second and third step of urine formation, that is reabsorption and secretion. Glomerular filtration has already been discussed in the class, so we are only taking two other steps here. Now, reabsorption. Reabsorption is different from absorption. Absorption happens in gastrointestinal tract, where after digestion, the basic finely divided nutrients are absorbed, whereas a reabsorption happens in the tubule of the nephron. As a result of filtration, almost all the components of the blood except blood corpuscles and major proteins get filtered, and that filtrate enters into the nephron tubule. So reabsorption is must to ensure that the essential parts of the blood are retained and conserved from being lost in urine. After filtration, reabsorption is the second basic function of the nephron and collecting duct. It is important to mention here that most of a reabsorption happens in proximal convoluted tubule. Now the slide here shows the routes of reabsorption. Reabsorption either happens between the cells, that's called paracellular reabsorption, or through cells, that is known as transcellular reabsorption. Solutes that are reabsorbed, that include glucose, amino acids, urea, and ions like sodium, potassium, calcium, chlorine, bicarbonate, and phosphate. Whole summary of reabsorption will be covered in the coming slides. The third and the most important function of the nephron is secretion. Secretion involves the transfer of materials from the blood and tubule cells into glomerular filtrate. That is the cell making the wall of the tubule. From there, some materials are secreted into the filtrate. The substances that are secreted include hydrogen ion, potassium ion, ammonium ion, creatinine, and certain drugs such as penicillin. Secretion leads to eliminating harmful things from blood and maintenance of blood pH, etc. This slide shows the summary of glomerular filtration, reabsorption, and secretion. Here you can see a reabsorption and secretion in proximal convoluted tubule is shown in yellow. Reabsorption and secretion in loop of Henle is shown in green, whereas reabsorption and secretion in DCT and collecting duct are shown in blue. Here you can also see the reabsorption and secretion of different substances in proximal convoluted tubule in this slide now. As already told, that a reabsorption of water, sodium, potassium, glucose, amino acids, chlorine, bicarbonate, urea, calcium, and magnesium happens. And it's already mentioned that maximum reabsorption happens in proximal convoluted tubule. What is the mode of reabsorption and how much reabsorption of different components happens in PCT? We will be seeing in coming slides. Here you can see also secretion of that is hydrogen ion, ammonium ion, urea, creatinine, all the secretion of these substances happen in PCT. Now you can see here the reabsorption of water, sodium, potassium, chlorine, bicarbonate, calcium, and magnesium happens in loop of Henle as well. The secretion of urea happens in loop of Henle. Here in this slide, you can see in the early part of DCT, the reabsorption of water, sodium, chlorine, calcium happens. And in late distal convoluted tubule, reabsorption of water, sodium, bicarbonate, and urea happens, whereas secretion of potassium and hydrogen ion happens here. That is late distal convoluted tubule as well as the collecting duct is concerned here. Now, what are the different modes of reabsorption? How does they happen? What happens? We will be seeing. This is the comparative reabsorption chart. What is the reabsorption in case of PCT and how it happens in case of loop of Henle, then early DCT or late DCT or collecting duct, they both are together in case of late DCT. Both reabsorption as well as secretion of different substances at different levels are through different modes that happens. This is a comparative chart. We will be discussing a little bit it in detail. But before we discuss what are the different modes of reabsorption and secretion, you can see different processes are responsible. That is osmosis, diffusion, facilitated diffusion, symport, antiport. It is that these different modes through which reabsorption or secretion happens. Before we go for the comparative chart, let's discuss here what is osmosis. Osmosis simply means diffusion of water from lower solute level to higher solute level. If the two solutions are separated by a membrane, the water will move from that solution where the solute concentration is low and water concentration is high to that very solution where solute concentration is high and water concentration is low. So we can say in osmosis, water moves from higher concentration to lower concentration in terms of water, from lower concentration to higher concentration in terms of solute. This is osmosis. Diffusion is actually the movement of substances across membrane from regions of higher concentration to lower concentration. This is diffusion. And for diffusion, there are no special channels, special proteins for taking the solute from one side of the membrane to the other. It is simply through the membrane. Now, another type of diffusion, that is facilitated diffusion. That diffusion which occurs through specific transmembrane proteins, that is called facilitated diffusion. That is diffusion, it's not at random through the membrane, rather the diffusion which happens through special proteins, gated proteins, this is called facilitated diffusion. And in facilitated diffusion we can see symport and antiport. In symport and antiport, two different solutes or molecules are transported together. When they are transported through a facilitated protein, facilitated channel, or facilitating that is gate, when the two molecules are transported in the same direction, it's called symport. When they are transported in different anti directions, that is one goes to one direction, another goes opposite to it, this is called antiport. These are two different types of facilitated diffusion. These are the different processes, that osmosis, diffusion, facilitated diffusion, all these happen in different parts of the tubule for ensuring the process of reabsorption and secretion. Now, here you can see the comparative slide. Reabsorption of water. The first substance that is to be taken into consideration is water. 65% of water is transported in PCT osmosis, that reabsorption happens. It comes from filtrate back into blood. Only 15% happens in loop of Henle, and then 10 to 15% happens in early DCT, and last 5 to 9% happens in late DCT or collecting duct. But it is important to mention here that in late DCT and collecting duct, the water reabsorption is dependent upon the ADH, anti-diuretic hormone. If ADH is there, water will be reabsorbed. If ADH is absent, no water will be reabsorbed. We will be discussing it later as well. Second is sodium. 65% sodium is symported or antiported through sodium potassium pumps. In case of PCT, 20 to 30% symportus in ascending limb, not in descending limb of loop of Henle. If you remember the structure of the nephron, there is descending loop of Henle and then ascending limb of loop of Henle. It is in ascending limb of loop of Henle that 20 to 30% of this sodium is reabsorbed in case of loop of Henle in ascending limb. 5% is symported in early DCT, and 1 to 4% in sodium potassium pumps or channels. They are stimulated by aldosterone only. This is conditional. Late DCT and collecting duct, the sodium will be reabsorbed. It is conditional if the it is required in body under the effect of that is aldosterone. Sodium will be reabsorbed, otherwise it will not be reabsorbed. Now the third is potassium, but first let's discuss glucose and amino acids. You can see glucose 100% reabsorbed in case of PCT. Similarly, amino acids 100% reabsorbed through symport and facilitated diffusion or symport and facilitated diffusion, both cases. It means when they are reabsorbed 100%, then there is no reabsorption in case of loop of Henle, early DCT, late DCT, or collecting duct. If small amount or minute amount of glucose is found in urine, it is said to be a defect, it is said to be a disease. Otherwise, 100% glucose or amino acids are reabsorbed in PCT. We already mentioned that it is maximum reabsorption that happens in PCT. Third one, if you see here, it is potassium. 65% is reabsorbed in PCT through diffusion. 20 to 30% it is symportus in ascending limb. Okay, then we say chlorine, 50% through diffusion in case of PCT, 35% symportus in ascending limb, and 5% through symportus in early DCT. Okay, then bicarbonate is similarly 80 to 90% facilitated diffusion in case of PCT, 10 to 20% through facilitated diffusion in case of loop of Henle, and if small amount, if it is present, the variable amount okay, it's all chlorine is all taken up to this early DCT level. Then there is bicarbonate, 80 to 90%. It is in PCT through facilitated diffusion, 10 to 20% through facilitated diffusion in case of loop of Henle. Then variable amount, it depends upon the concentration. It depends upon the H positive secretion. If more H positive is secreted, then bicarbonate will be taken into in late DCT and collecting duct. Urea 50% through diffusion in case of PCT, it is reabsorbed, and then later on in late PCT or collecting duct, variable amount is reabsorbed. It depends upon the conditions, and it is cycled to nephron loop to maintain the osmotic gradient. We will be seeing how this happens later on. Similarly, calcium and magnesium, it is variable amount in case of PCT through diffusion, variable amount in case of loop of Henle through diffusion. To especially calcium, variable amount and is stimulated by parathyroid hormone PTH in case of early DCT. It is important, not magnesium, only calcium, and it is stimulated by PTH. These are the things reabsorbed and where they are reabsorbed, what is the mode, how much percent they are reabsorbed. It is given in comparative chart here. Let me again mention here that late DCT includes collecting duct as well. Okay, now the secretion is mainly of H positive, NH, that is ammonia, NH4, urea, creatinine, and similarly K positive as well. H positive, you see, in case of PCT, H positive secretion, it is variable. There is no fixed percentage, and it happens through antiportus. And then later on in late DCT or collecting duct, variable amount to maintain the acid base homeostasis in the body. H positive pumps secrete H positive. Okay. Similarly, ammonia, if you see, variable amount is secreted in case of PCT only. Ammonia. Variable amount to adjust the dietary intake of potassium. What is the level of potassium? How much it is to be secreted? It depends upon the homeostasis of the body. And potassium is secreted in late DCT. Okay. In the row of ammonia, it is potassium given in late DCT case. It is not ammonia. Similarly, urea diffuses in variable amount in PCT. Similarly, it is creatinine, small amount is secreted in case of PCT, and no amount is secreted in case of late DCT, early DCT, or loop of Henle. Okay, these are the different conditions in which the reabsorption and secretion happens in loop of Henle. Okay, after final filtration, reabsorption and secretion, the product that is formed, it's known as urine. Urine, now it can be dilute or concentrated. It depends upon the conditions or the status of water in the body. If water in concentration in body is extra and there is no need to conserve it, dilute urine is produced. On the other hand, when water intake is low and water loss is high, such as during heavy sweating, the kidneys must conserve water while still eliminating wastes and excess ions. For that, urine is needed. Urine is need to be produced in these conditions because extra these ions they are to be secreted. Now, under the influence of anti-diuretic hormone, kidneys produces small amount of highly concentrated urine. That is up to 4 times more concentrated than blood plasma. The osmotic concentration of plasma at the blood is generally around 300 milliosmoles per liter, but the concentrated urine can be up to 1200 milliosmoles per liter. That's 4 times. The ability of anti-diuretic hormone to produce highly concentrated urine. It depends upon the presence of osmotic gradient of solutes to interstitial fluid of renal medulla. Three major solutes that contribute to osmotic gradient, they are sodium, chlorine, and urea. We have not discussed here the formation of dilute urine. We will be rather focusing on the formation of concentrated urine, and the process itself will make clear to you people how dilute urine will be formed. The main factors that are responsible for the formation of osmotic gradient in renal medulla. There are two main factors. One is difference in solute and water permeability in different regions of the nephron tubule, and the second one is counter current mechanism. If we see the difference in water and solute permeability in different regions of the nephron, we can see that the descending limb of loop of Henle is permeable to water only. It does not allow the permeability of ions except urea. The ascending limb of loop of Henle is permeable to ions only. It does not allow water. The early part of the DCT, distal convoluted tubule, can allow water to some extent as well as ions. The late part of DCT as well as collecting duct, they allow the water permeability under conditions that is conditional. That is early part of DCT. No, it is late part of DCT and collecting duct. They allow water permeability under the effect of anti-diuretic hormone and permeability to sodium under the effect of aldosterone. It is conditional. Now, countercurrent mechanism, it involves countercurrent multiplication and countercurrent exchange. What is this countercurrent multiplication? Let's understand. Countercurrent multiplication, actually it is the formation of osmotic gradient. Uh, it forms progressively increasing osmotic gradient in interstitial fluid as a result of countercurrent flow. Different steps that are involved in it is, the first one is the symptotus in thick ascending limb cells of nephron loop cause a buildup of sodium and chlorine in the renal medulla. जो ascending limb of loop of Henle होता है, उसके अंदर सोडियम एंड क्लोरीन सिम्पोटस मौजूद होते हैं। यानी दिस वेरी पार्ट ऑफ द नेफ्रॉन, ये इम्पर्मिएबल टू वाटर, ये वाटर को अलो नहीं करता है, सोडियम एंड क्लोरीन को अलो करता है बाहर निकलने के लिए। तो जब सोडियम एंड क्लोरीन बाहर निकलते हैं तो आउटर साइड में इंटरस्टिशियल मेडुला के अंदर, रिनल मेडुला के अंदर बिल्डअप ऑफ सोडियम एंड क्लोरीन हो जाता है, जिससे ऑस्मोलरिटी बढ़ जाती है। सेकंड स्टेप और सेकंड फैक्टर इसमें आप देखेंगे, काउंटर करंट फ्लो थ्रू द डिसेंडिंग एंड असेंडिंग लिम्ब ऑफ द नेफ्रॉन लूप एस्टैब्लिशेस एन ऑस्मोटिक ग्रेडियंट इन द रिनल मेडुला। ये कैसे होता है? जो डिसेंडिंग लिम्ब ऑफ लूप ऑफ हेनले है, दैट अलाउज वाटर टू कम आउट, क्योंकि वाटर के बाहर ऑस्मोटिक ग्रेडियंट है, सॉरी ये लूप ऑफ हेनले के बाहर ऑस्मोटिक ग्रेडियंट है। तो डिसेंडिंग लिम्ब वाटर को एलो करेगी, क्योंकि वाटर परमिएबल है, वो आयंस को एलो नहीं करेगी। तो ये फिल्ट्रेट जब असेंडिंग लिम्ब के पास पहुंचेगा, तो असेंडिंग लिम्ब चूंकि वाटर निकल चुका है तो टॉनिसिटी ऑलरेडी हाई है, तो जब असेंडिंग लिम्ब के पास पहुंचो तो वहाँ आयंस को एलो किया जाता है, वाटर्स को एलो नहीं किया जाता। तो वहाँ पे फिर आयंस बाहर आ जाएंगे, तो वाटर नहीं आएगा। ठीक है, तो इस तरह से ये डिफरेंशियल परमिएबिलिटी टू आयंस एंड वाटर, इससे ऑस्मोटिक ग्रेडियंट जनरेट होता है, क्योंकि डिसेंडिंग लिम्ब वाटर को एलो करेगी बाहर निकलना, क्योंकि बाहर आयंस हैं तो वाटर बाहर आएगा। असेंडिंग लिम्ब आयंस को बाहर करेंगे, क्योंकि असेंडिंग लिम्ब तक पहुंचते पहुंचते फिल्ट्रेट के अंदर आयंस की कंसंट्रेशन ज़्यादा हो जाती है, क्योंकि वाटर निकल चुका है डिसेंडिंग लिम्ब में, तो वहाँ से फिर आयंस बाहर आ जाते हैं। तो इस तरह से एक साइड से वाटर का निकलना और अंदर टॉनिसिटी का बढ़ना फिल्ट्रेट की, और फिर आयंस का बाहर आना। तो इस तरह से क्या होता है, ये काउंटर करंट फ्लो से इट रिजल्ट्स इन द जनरेशन ऑफ progressively increasing osmotic gradient in the interstitial fluid. थर्ड फैक्टर इसमें क्या है, cells in the collecting duct absorb more water and urea. ADH के इफेक्ट से, ADH की प्रेजेंस से the late part of collecting duct and the late part of DCT, sorry, the late part of DCT and collecting duct, under the effect of ADH it becomes permeable to water. तो वाटर जो वहाँ से ले लिया जाता है थ्रू द एजेंसी ऑफ प्रिंसिपल सेल्स, तो जब वाटर लॉस वहाँ से हो जाता है तो वहाँ फिर टॉनिसिटी इंक्रीज हो जाती है, तो उससे फिर यूरिया भी बाहर आ जाता है। In the absence of ADH, no water is reabsorbed in late DCT and collecting duct. Whereas when ADH concentration is there, it is the late DCT and collecting duct, they are permeable to water, and water is reabsorbed through principal cells. And when the tonicity of the fluid inside increases, because the area is also permeable to urea also diffuses out from the region of late DCT and collecting duct. The fourth factor is urea recycling, and this urea recycling causes the buildup of urea in the renal medulla. How? नेफ्रॉन की डिसेंडिंग लिम्ब ऑफ लूप ऑफ हेनले, थिन असेंडिंग लिम्ब ऑफ लूप ऑफ हेनले, और कलेक्टिंग डक्ट का वो पोर्शन जो मेडुलरी रीजन के अंदर है, दीज आर द रीजन्स विच आर परमिएबल टू यूरिया। इसके बरक्स अगर आप थिक असेंडिंग लिम्ब और अर्ली पार्ट ऑफ डीसीटी एंड द पार्ट ऑफ डीसीटी एज वेल एज कलेक्टिंग डक्ट इन कॉर्टिकल रीजन के अंदर हमने क्या कहा, थिक असेंडिंग लिम्ब, the DCT as well as the collecting duct in cortical region, these are impermeable to urea. तो जब यूरिया मेडुलरी रीजन के अंदर कलेक्टिंग डक्ट के अंदर होता है तो वहाँ चूंकि टॉनिसिटी काफ़ी हाई हो जाती है तो यूरिया बाहर डिफ्यूज़ हो जाता है। फ्रॉम दियर दिस यूरिया गेट्स शिफ्ट टू द renal medulla where it enters to the thin descending as well as thin ascending limb of loop of Henle, because these are the regions which are permeable to urea. तो वहाँ से यूरिया फिर इस फिल्ट्रेट के अंदर थ्रू DCT collecting duct it again reaches to the part of collecting duct in medullary region, where from it again gets recycled back to the loop of Henle portions. And in this way urea recycling happens, and this urea recycling leads to the build-up of osmotic gradient in case of renal medulla up to 1200 milliosmoles per liter. This osmotic gradient, this high rate of osmotic gradient or high value of osmotic gradient is created at the lower tip of the medullary nephron actually. Second important factor for the maintenance of osmotic gradient is counter current exchange. And the counter current exchange is actually the process by which solutes and water are passively exchanged between the blood and the vasa recta and interstitial fluid of the renal medulla as a result of counter current flow. काउंटर करंट फ्लो जिस तरह से रेनल लूप ऑफ हेनले के अंदर होता है, इसी तरह से काउंटर करंट फ्लो वासा रेक्टा के अंदर भी होता है। तो वासा रेक्टा के अंदर जब ब्लड एक लूप में नीचे आता है और दूसरी लूप में ऊपर जाता है तो काउंटर करंट फ्लो होता है। तो जब लूप में डिसेंडिंग लूप के अंदर ब्लड जब नीचे आ रहा होता है तो बाहर की टॉनिसिटी ज़्यादा होने से उसमें से वाटर लॉस हो जाता है, नीचे आते आते उसमें से वाटर लॉस हो जाता है और इस तरह से जो ब्लड अंदर बचता है उसके अंदर आयन ज़्यादा बचते हैं और वाटर कम बचता है। लेकिन जब ब्लड असेंडिंग लिम्ब से ऊपर आ रहा होता है तो उस वक्त चूँकि उसमें आयन ज़्यादा होते हैं, वाटर कम होता है, तो वहाँ से फिर वाटर दोबारा से इसमें चूँकि परमिएबल है, वाटर दोबारा से अंदर आ जाता है और आयन्स लॉस हो जाते हैं और इस तरह से फिर ब्लड निकल जाता है। तो ये जो काउंटर करंट फ्लो वासा रेक्टा के अंदर है, इट इज़ नॉट एक्चुअली क्रिएटिंग और डिक्रीजिंग दिस ऑस्मोटिक ग्रेडियंट। इसका रोल सिर्फ है मेंटेनेंस ऑफ ऑस्मोटिक ग्रेडियंट। द ऑस्मोटिक ग्रेडियंट दैट हैज़ बीन जनरेटेड दैट इज एक्चुअली मेंटेन्ड continuously with the help of vasa recta, and this is called counter current exchange because it is the exchange of these ions as well as water with the blood that results in the maintenance of this osmotic gradient. This was all about the last two steps of the urine formation, that is reabsorption and secretion, their summary, and finally the formation of dilute urine as well as the concentrated urine. And let me remind once again, the dilute urine will be formed when water will be in huge quantity inside the body and there will be no more need to consume the water. So under the effect of ADH, no water will be. Sorry, in the absence of ADH, urine is very dilute. However, very high level of ADH stimulates reabsorption of more water into blood and thus that produces a concentrated urine. Thank you. Thank you very much.